So when we are looking at this, we're going to start by kind of doing a bit of review of the things that we talked about on Tuesday and then continuing on. So the first thing that we talked about, if we zoom in here on the side, okay, the first thing that we looked at was we just looked at the cell membrane and the ways that what that cell membrane is composed of, as well as how transport happens. So when we were doing that, we looked, again, you'll see my drawing abilities. So we looked at the molecule that makes up the phospholipid bilayer, which was our first main term. So just a reminder, that phospholipid bilayer is the same as the cell membrane. Okay, so the phospholipid bilayer is both in plants and animal cells, as well as bacteria cells. Every single cell has a phospholipid bilayer. We just call it the cell membrane sometimes when we're feeling lazy or we don't want to use the science term, but that science term is the phospholipid bilayer. And the reason it's called that phospholipid bilayer, again, is because it has a phosphate head as well as two lipid tails. Okay. So each and every molecule in the cell or the phospholipid bilayer is made up of that one phosphate and two lipids. And the reason that we talked about that was important was because the phosphate is hydrophilic, whereas the lipids are hydrophobic. Again, the reason that was important was because it means that something that is either a small lipid or is also hydrophobic can pass through the phosphate group and move through the lipid layer into the cell easily without restriction. Whereas if it is something that is hydrophilic, so it likes water or would normally dissolve in water, or is water itself, it can't freely move through the cell membrane. And so the structure of that phospholipid is actually very important in that because it tells us what is allowed into the cell easily and what needs help getting into the cell. Okay, so that was the first thing that you needed to know, was what that phospholipid looks like, what it's composed of, and how it restricts certain molecules from coming in and out of the cell. Okay, then again, breezing on through to the things that we talked about last day. So we looked in this first section of the diagram and we talked about diffusion. Okay. We talked about how molecules can move from an area of high concentration, high to low concentration. Again, with the model of the tea bag getting put into water. So if you put that tea bag into a hot cup of water, the tea is going to move from where there's a high concentration of tea to a low concentration of tea until the entire cup is filled with that tea beverage. Okay, and so the molecules that we talked about that can move freely through diffusion, so don't need help from a protein or anything like that, we talked about oxygen, carbon dioxide, as well as small lipid molecules. Okay, and we showed that they could go into and out of the cell with a double-ended arrow, just showing that they can go in and out freely to fit whatever is a even concentration throughout. Okay, so that was the first thing that we talked about. Again, this is just review, so these things should feel fairly familiar, but I think it's worth going through again. Okay, the next one that we looked at was this second from the left. And so this we said was a protein channel. Okay, remembering that in our diagram, it often will look like this kind of set of three rectangles squished together. But in reality, when you see this in a cell membrane, it'll look like a straw poked through the cell membrane. And it just creates that space between the lipids so that hydrophilic molecules or water-loving molecules can pass through more freely. Okay. Again, these have to be pretty small molecules because the straw opening is not huge. So kind of the analogy we went through last time was you can't shove a blueberry through your smoothie straw. So it has to be a somewhat small molecule in order for it to be able to pass through. Oh, don't know why that's jumping around. So the molecules that we talked about that can go through this protein channel were your ions. So I'm writing the ions down here for the protein channel, but anytime later that I talk about ions, it's the same group of ions that can go in and out of the other proteins. But I don't want to write out them every single time. So when you see ions later on, it just means the same grouping of 4 ions. So those are sodium, calcium, chlorine, and potassium, and then water as well. Okay. And the title for how these molecules can move in and out of the cell is through facilitated diffusion. Okay, so again, the same property of diffusion where it can move from a high concentration to a low concentration until the concentration is even. And so again, when we're showing that, we show a double-ended arrow showing that they're able to freely move in and out based on whatever the concentration is. Okay, so they're not restricted by anything. They can freely go through the tube to either direction based on what that concentration is. Okay, the next one that we talked about was this one here, which is everything's moving around on me. Okay, which is our gated protein channel. So it's kind of like the protein channel with an extra step. And the important thing about this gated protein channel is that now the ions or the molecules aren't able to freely move in and out. There's a restriction or a step that they have to go through first. And so that restriction or that step comes through the protein or the gated protein channel itself. Okay, so the molecules that you're going to be looking at here are going to be, again, those same ions that we looked at before. So sodium, calcium, chloride, and potassium. But here I'm just going to write ions. Okay, as well as your amino acids and your sugar molecules. Okay, and if you remember from Tuesday's class, the thing that we talked about here is that those molecules are going to enter in whichever side is open. So traditionally we have the open side towards the extracellular space or towards the top of the page, but it can be open on the bottom as well. It's a still picture, still diagram, so I can't show it both ways, but it can totally be open on either end to let these molecules in. So just something to note. So that molecule is going to enter the gated protein channel here, and it's going to temporarily bind to the protein to show that it has entered the protein. When it temporarily binds and says hello to the protein then is going to switch its orientation or switch its position, and it's actually going to close at the top. So we can draw some arrows to show that it's closing at the top, and then it's going to open up at the bottom so that that sugar can flow through into the cell. Okay, again, could be exactly the same thing in reverse. So the sugar could be leaving the cell, and so the open end would be at the bottom. It would flow in, bind to the protein temporarily. The protein would change shape, and then it would be able to flow out. Okay. And we also talked about how this is facilitated diffusion. Okay. So here, this is where I'm going to pause, and I'm actually going to give you a new term. So when we are looking at, come on, technology, work for me. Okay, we'll do it that way. Okay, so when we are going to look at these 3 types of diffusion, so traditional diffusion, where the molecules just move in and out freely, facilitated diffusion through a protein channel, or facilitated diffusion through a gated protein channel, here we are looking at passive transport. Okay, so the term that we give to diffusion or osmosis, which we're going to talk about in a minute, is passive transport. So it doesn't require any effort from the cell. It happens pretty freely, where the ions can move in and out based on where the concentration gradient is, so from high to low. So the important thing with passive transport is it is always from a high concentration to a low concentration, every time. So all three of those, if it has the word diffusion in it, it is a type of passive transport, which means that it always goes from high to low concentration. Okay. Okay, moving on to our next protein, which is not actually a form of transport. So it's not passive transport or active transport, which we're getting into next. So this one is just a protein that is attached into the cell membrane. So it is bound within the cell membrane. It doesn't cross over from one side to the other. It's bound within it itself. And so this one we gave the name the integral monotopic protein. Okay. So again, this protein does not have anything to do with transport. It's just a protein that is within the membrane itself, but it is important to know about and that it exists in the cell membrane. So this integral monotopic protein has two main jobs that it could do, one or the other, which all come down to this cholesterol tail. Cholesterol tail. Okay.
That is not passive transport. That's just its own thing?
Yeah, this is its own thing. So it doesn't help with moving anything in or out of the cell. This is just its own thing entirely. Great question. It's just a part of the cell membrane. Okay. And the two main jobs that we talked about with this integral protein that it could have, so it's again, one or the other, not both, is it either marks or identifies the cell, identifies the type of cell. So all of your lung cells will have the same cholesterol tail with the same number of cholesterol in it, in the same orientation, whereas your heart cells will have also a cholesterol tail, but they'll be in a slightly different orientation or a different amount of cholesterol to mark that it is actually a heart cell and not a lung cell, and so on. Okay, so it either identifies that type of cell or its other job that it could do. Oh, come on. Is it can hold the flagella or cilia. Okay, so again, flagella is kind of like a tail, whereas cilia are kind of like feet. So flagella just helps the cell move around, kind of like a fish's tail in water. That tail helps it move in one direction. Generally, a cell will either have one flagella or two, but not generally more than that. Whereas cilia, usually kind of around the entire cell, you'll have these cholesterol tails, which form cilia, and so you'll have a bunch of them that'll surround the tail, and those cilia help it move, usually quite quickly. Okay, so again, flagella, usually one or two per cell, whereas cilia can be hundreds per cell. Just depends on what the cell needs to do, and those are usually seen in bacteria. Both of these are usually seen in bacteria. Okay, you don't often see those in the human body. Right, so that is the recap of everything that we talked about on Tuesday. So I'm going to pause there. Do you have any questions so far? Rachel, you have a question?
Just going back to the gated protein channel, you said that typically the entrance is on the extracellular side?
Yeah, so typically it's on the extracellular side, but it can be on either.
Okay.
Yeah. Great. Any other questions? No? All right. So again, you can always type them into the chat if they come up as we go. Or just feel free to pause me. So this is our new material that we're going to be getting into. So we are now going to look at our final protein that is going to be in the cell membrane. So again, remember that this is a transmembrane protein. So it crosses the entire length of the cell membrane. So it opens at the top and closes at the bottom, or vice versa. And so it spans that entire length, unlike the integral protein, which only enters at one side. So this one, we are going to title active transport. Okay, so remember the last three types of transport that we talked about being diffusion and facilitated diffusion. Those are both passive transport, so they don't require any energy input from the cell. They just happen through the ions moving along their concentration gradient. Whereas active transport requires energy from the cell. In order for that transport to happen. So active transport, again, requires energy from the cell in order for this type of transport to happen. And the reason for that is because now you are moving from a low concentration of solute to a high concentration. Okay, so you're moving it against its natural flow. So molecules want to move in a diffusion process where they're moving from a high concentration to a low concentration because they want to spread out. They want to have as much space as they can. So they like to spread out. That's their natural way of moving around. So with active transport, we go against that natural tendency that those molecules want to do, and we force them to actually get closer together. So it's like you're cramming a bunch of molecules close together that don't really want to be close together. And so that requires that energy from the cell in order to make that happen, to force them to get closer together than they would like to. Okay, so that's why we use that energy from the cell. That energy comes from a molecule called ATP, okay, or adenosine triphosphate, if you want to use the fancy term. You just need to know that it's called ATP. Okay, so it is an adenosine molecule that is bound to 3 phosphate groups. Okay, and those 3 phosphate groups, this is the chemistry behind it if you want to know. So those 3 phosphate groups that are bound to that adenosine, in those bonds between the adenosine and the phosphate, they hold a lot of energy. And so when we break the connection between the adenosine and the phosphate, that releases a bunch of energy. And so what the cell does is, if you see this little lightning bolt, that's the energy being put in. So that's your ATP. That's just freely in the cell. Okay, and this protein here is going to cause that adenosine triphosphate to break apart and split it into an adenosine diphosphate plus a phosphate that will now just free float around. Okay, and it's that breaking up of that adenosine triphosphate, or that ATP, into ADP that actually releases the energy and frees it so that this protein can move those molecules against their concentration gradient, or against the way that they would normally like to go, forcing them together. Okay, so that's where the energy comes from. So from ATP, the ATP is broken up into ADP, and that releases that energy because that bond is broken. Hopefully you maybe remember that from chemistry a little bit. Okay, and so what you're going to see with this one, traditionally you're going to see sodium and potassium talked about here. Okay, So generally, you're going to be seeing sodium and potassium moving through this in the sodium-potassium pump, which is a very significant one that you're going to be looking at in your textbook, actually. But we're going to talk about it a little bit now as well. So we're looking at those two. We can also see sugar moving through this channel. Okay, also amino acids. And occasionally water. Okay, so again, those groups are all part of here and what can be moved there. Again, sodium and potassium being the usual ones that will transport through there, but the sugar and amino acids, etc., can also be moved through this molecule. It works very similar to the gated protein channel, where the molecule will move in. So let's say we're looking at potassium. Okay, so that potassium will move in. It will temporarily bind to the protein. In this protein, when it senses that that potassium has bound there, then that's when that ATP to ADP reaction happens, or breakdown happens to release that energy, and that forces this protein to change its shape. The protein wouldn't normally change its shape with that action or with that energy being put in. It forces that change of shape for this protein, allowing the potassium out. So again, without that energy input from the cell, the potassium wouldn't be able to move across. It would be stuck on top. Okay, remembering that this is our one low to high concentration move of those ions. Okay. So I'm going to pause there again. Do we have any questions? We're good? Okay. So moving on, we have two more things we're going to be talking about. Technology. Okay, so we are going to be now looking at osmosis. Okay, so osmosis is a type of cell membrane transport that happens in protein channels. Okay, so this is all going to be about osmosis, which happens through protein channels. Which, when you hear the term protein channel, that should ding in your mind the memory that that is a type of passive transport. Okay, so it doesn't require energy to move through a protein channel. Okay, it is also facilitated diffusion. Okay, again, that facilitated doesn't mean energy, it means the use of a protein. Okay, so the protein helps open up that cell membrane a little bit to let in your hydrophilic molecules. Okay, and when we're talking about osmosis, we're specifically talking about the transport of water. Okay, so when we are looking at osmosis, the only type of molecule we're looking at is the transport of water. Nothing else moves via osmosis. Okay, and the last important thing to know from this, with that facilitated diffusion, that should put in mind that we are moving from a high concentration of water to a low concentration of water. So again, the water wants to spread out take up as much space as it can, and so it's going to move from where there is a lot of water molecules crammed together to where there is no water molecules crammed together. Okay, when we're looking at this osmosis, we're going to look at 3 different scenarios of what will happen to a cell in 3 different types of solution. So solution being the liquid that a cell is put into or surrounded by. Okay, so we're going to look at 3 different types of solution. One that has a high concentration of ions in it, one that has a medium amount of ions in it, and one that has no ions in it, and the effect that that will actually have on the cell and the movement of that water across the cell membrane. Okay, so the first one we're going to look at, oh, come on. Haha. Okay, the first one we're going to look at is we are going to look at a hypertonic solution. Okay, hypertonic solution meaning that there is a high concentration of solute or salt in the liquid. Okay, that could be water, could be any liquid. Okay, so hypertonic solution means that there's a high concentration of solute or salts in the liquid. And so when we're looking at that, zoom in again, there we go. Okay, this is your water in your beaker. So this you might want to draw on the back of your page or on a separate sheet of paper, but this is linked to the cell membrane transport. Okay, then if you have your cell, that has been plopped into the water. This is a very large cell. Okay, so you have your cell that's been plopped into the water, and in this water solution, there are obviously water molecules. But in a hypertonic solution, it's going to be very salty. So you're going to have a lot of sodium and chloride floating through. Okay, so they're kind of crowding out the space for the water molecules. Okay. So there are more ions than there are water molecules, or there's a higher concentration there. Whereas inside the cell, there's also water. Okay, and there are also always salts within the cell as well. Okay, but in this case, where it's dropped into very salty water, these water molecules inside the cell are more crammed together than the water molecules on the outside of the cell. So outside of the cell, if you look at the water molecules, though there is still water there, they have a lot more space because they're kept separate by those salt ions. Whereas inside the cell, the water molecules are actually shoved closer together by the cell membrane because it's crammed closer together. So the water molecules inside the cell actually have less space than the water molecules on the outside. So there's a higher concentration of water. Ooh, thick pen. Okay, so your concentration of water specifically is higher inside the cell than outside of the cell. So again, with the diffusion or the osmosis of water, the water is going to move from a high concentration to a low concentration. So all of these water molecules are actually going to move out of the cell. So they can spread out as much as they can into the cell. So what that means is that our cell, after this happens, is going to shrink up. Here's my shrunken cell. Okay, so the cell is going to shrink because it's becoming dehydrated, because all of the water is leaving, and all that's left inside is those salt ions. Okay, so it's going to shrink down because the water has left and moved out. Okay, so the cell shrinks. Another word that we use for that is called plasmolysis. Okay, plasmolysis just means shrinking. Okay, so the cell shrinks or plasmolized. Okay, so again, if a cell is put into a very high solute concentration liquid, the water molecules inside the cell are going to move out or to have more space. So they're going to leave the cell and the cell is going to shrink. Okay. So then we're actually going to shift over to our last beaker and look at the opposite end of the spectrum. Okay, so hypertonic solution, the cell is going to shrink or plasmolyze. Whereas here, we're going to look at a hypotonic solution. Okay, that hypotonic solution is going to be one that has little to no salt concentration. Okay, so think of distilled water, for example. So you pretty much have only water molecules in that solution. So again, if we draw our liquid is in there, and we have our cell. Okay, so you're going to have pretty much only water molecules in our beaker, taking up all the space. Okay, again, remembering that inside the cell there are still those water molecules exist inside there, and you have inside the cell you always have, inside the cell you also always have those salts as well. Those are always going to be inside the cell, taking up space. So in this example, the water on the outside of the cell is a much higher concentration. There's much more water molecules on the outside of the cell versus on the inside of the cell. So again, the water molecules are always going to move down their concentration gradient, so from a high concentration to a low concentration. So in this scenario, what that looks like is it's going to move into the cell. So all of your water molecules, not all, but a lot of your water molecules are going to move inside the cell. Okay. Because they want to spread out. And what that looks like for your cell is your cell is going to expand. So it's going to become much larger. So cell swells or expands. Okay. Another word that we can use for that is it lyses. Lyse means to swell. Okay. And if in some scenarios, if the water molecules are concentrated enough, it can actually cause it to explode. Okay, so sometimes the cell will actually explode and the cell will die. Same as in the hypertonic solution, if too much water rushes out of the cell could shrink so much that it actually dies as well. So either of these scenarios can actually be deadly to the cell. Okay, and the last one we're going to look at is our nice mid-ground one, okay, which is an isotonic solution. Oh no! Scroll down here. Come on. Always gotta have one lapse of technology. Okay, so we're gonna look at our middle solution. So this one is going to be an isotonic solution. Okay. Again, isotonic is kind of the middle between your hypertonic and your hypotonic solutions. So it's going to have some solutes dissolved in the water, not nearly as much as the hypertonic and more than the hypotonic solutions. Okay, and this is where your cell is happiest. It wants to be in an isotonic solution. That's how it's going to do best. So again, if you have your cell, my cell keeps changing size depending on the solution it's in. But if that's your cell, so again, you have those water molecules on the outside. And you have your salts, but only a few this time. Okay. Inside the cell, you again have those same solutes, as well as water. Okay. And if it's balanced, the amount of solutes outside of the cell and inside of the cell, then the water is actually going to freely move in and out. Okay, so it's just going to flow in and out of the cell, and the cell's going to stay the same size. Okay, so again, reminder with this that we are just talking about the water molecules and where they are going to be moving. But it's important to keep in mind that the ions can move in and out of the cell. If you remember back to the passive and active transports, those ions can move through protein channels, through gated channels, as well as through your active transport carrier proteins. Okay, so those ions can actually move in and out of the cell, and the cell actually has some control over how those ions move a little bit. And so if the cell is dropped into a hypertonic solution, it has a little bit of wiggle room for being able to adapt to that hypertonic solution or the hypotonic solution. It has that ability to adapt. But if we're looking at a very, very salty water solution, the cell can only do so much, and so it still will shrink and eventually shrivel up and die if it's too hypertonic. So just a reminder with that the cell does have some ability to control those ions and help itself out with that. But in osmosis or in these diagrams, we're just looking at the water molecules and how the water molecules would move if the cell did nothing else. Okay. Any questions on osmosis before we move on to our last transport? No questions so far? Great. So the last way that your cell has for moving molecules in and out of the cell, this one is specifically for large molecules. Okay, so everything we've been looking at so far is individual ions, small glucose molecules, amino acids, so very tiny molecules. So now we want to look at some larger molecules. Can we pause for a minute? Yes, Marissa, we can pause for a minute. I know you're probably all frantically writing. But again, remember, I am recording this, so you can go back and revisit it. Look at all my technology fails again. Do you still need another minute? Okay, great. So we will move on unless anybody frantically types in that they need another.
What are we waiting for?
Some people just needed a second to catch up. Lots of information.
What?
All right, great. Okay, so the last one that we're going to look at is the transport of those large molecules in and out of the cell. So here we're looking specifically at long chain proteins. Okay, so remember that all of your proteins are made in the ribosome of the cell. And so after those proteins are made, they're not always needed within that same cell that makes them, and sometimes they need to be transported to other cells or other places that are going to use that protein. And so we need a way to get that protein out of the cell without completely punching a hole through the cell membrane. Again, the protein would not be able to fit in between the tiny little phosphates. And we don't want to create too much of a gap because then everything inside the cell would rush out. And so the cell had to come up with a creative way around that problem of moving those proteins out of the cell, as well as bringing some larger molecules in. So sometimes it wants to bring proteins into that cell so that it can use them or even break them up. And sometimes it needs to get rid of large waste molecules that it doesn't need. So it just needs to get rid of a bunch of molecules all at once. So it's going to go through either these two processes to take something in or to get rid of something, move something out of the cell. So the first one that we're going to look at is we are going to look at endocytosis. Okay, endocytosis. So taking large molecules into the cell. So from the extracellular space to the intracellular space, if you remember those fancy terms. Okay. So again, what that looks like, if I draw a nice curved shape. So this is going to be the curve of your cell, okay, part of your cell membrane. So again, remembering your cell membrane isn't just a straight line, it's actually made up of all of those tiny little phosphate lipid groups. Okay, so all the way around, you're going to have those tiny phospholipids. Okay, and what your cell is going to do is if there's a large protein that comes up to the cell and wants to enter the cell, it's going to brush up against the cell and temporarily bind to those phosphate groups themselves. So similar to the gated protein channels where the glucose was temporarily binding to that protein, the protein here that needs to enter the cell is going to temporarily bind to the cell membrane. Okay, so it's going to attach on, it's going to latch on, and the cell membrane, what it's going to do is it's going to suck in that protein and actually pinch off part of the cell membrane, closing it up, but also encasing that protein in a little bit of cell membrane. So what that looks like is if you have your protein here that has now attached, PR, let's say, for protein. Okay, so this cell membrane is going to kind of form a little pocket that protein is going to slowly go into. Again, the protein is temporarily binded there, so it's stuck onto the cell membrane. And as it moves along, that pocket is going to get larger and larger. Okay, and eventually bind off. Okay, and then these phosphates at the top here of the cell membrane that were quite far apart beforehand are now going to be really close together, and they're going to bind and pinch off that bit of the cell membrane, closing it again, so there's not any gap at the end. So it's like the cell membrane kind of like shuffles and gets a little bit smaller to create that that ring of phospholipids around the protein. So now it's kind of like a little carrier, or a little vesicle, it's called, with the protein inside. And then that vesicle, or the little carrier of phospholipids, is going to carry that protein to wherever in the cell it needs to go. Okay, so this is endocytosis. So large molecules entering the cell, where the cell membrane pinches off and envelops the protein, carrying it into the cell and wherever it needs to go. Okay. And exocytosis, moving on, is exactly the opposite. Okay, so exocytosis, we're going to be looking at large molecules leaving the cell. Okay, so this would be proteins that have just been synthesized and need to be used by another cell, or in a different space, or removing large amounts of waste products that the cell has created that it needs to get rid of really quickly, instead of just letting those wastes naturally flow out of the transport channels or the gated protein channels. This just kind of lets it get rid of a lot all at once through exocytosis. So again, same idea. So you have your cell membrane, you have your vesicle that is going to have a bunch of those molecules or the protein inside of it. Okay, so when that protein is synthesized, it kind of takes over one of these vesicles, which is just going to be flowing, or they're just going to be kind of around in the cell, empty, and then the protein will be put inside of it to be transported out of the cell. So again, that vesicle is made up of all of those phospholipid ring all the way around. Okay, so it's like a little bit of cell membrane. And so what it's going to do is it's going to do exactly the opposite. So it's going to move up towards the cell membrane and attach on there. So you have your protein that attaches, or the protein vesicle that attaches to the cell membrane, and then it's going to kind of split open the cell membrane as this vesicle kind of unfurls almost from its attaching point. Oh, not what I wanted. Okay. Once it attaches and sort of starts to unfurl and spread out that cell membrane, then the protein can leave. And that vesicle phospholipids that are attached there, they remain attached. So this is how the cell doesn't just keep shrinking and shrinking and shrinking over time through endocytosis as that is being pinched off. Here it's being replaced, or the cell membrane is spreading out again to kind of keep or maintain the same amount of cell membrane as we had before. So it'll attach on there and then spread out to create that nice smooth phospholipid bilayer along the outside.